Chronic wasting disease is an animal disease that affects primarily deer here in the Midwest and across the country. It is a disease that affects the brain of the deer, and it causes them to essentially waste away and die very quickly. The research that we are doing is using composting as a very quick and easy and practical means to basically get rid of chronic waste disease infected deer and other cervids here in Wisconsin and the Midwest. We are taking infected carcasses. We are composting them at a quarantine site here in central Wisconsin where there's no public access, and we are following through the composting process using a soil layer, using sawdust as our carbon source, using infected carcasses, and essentially monitoring the composting process over the course of 4 to 6 months to see whether or not the infectious prion, or the disease-causing agent for chronic wasting disease, is actually being eliminated, degraded, destroyed, or whether or not it's persisting in the compost pile or moving out of the compost pile.
We're using a technique called biopiling. So we're using a heavy amount of carbon to offset the heavy amount of nutrients and nitrogen that necessarily comes from the carcasses. And we're putting those two together in a ratio that creates a composting cycle. We're getting high temperatures and creating a high environment for heavy microbial activity, and those microbes turn those carcasses and that carbon material into compost.
We are taking samples of our compost material. We're testing the deer before they go in to begin with. We're taking soil samples, we're taking sawdust samples, we're collecting effluent from the cells, and then we're testing that through the Joel Peterson lab at the University of Wisconsin-Madison, where as part of this project, they are perfecting their RT-Quick method for how prions are detected in environmental samples.
They're using a method called RT-Quick, which stands for real-time quaking-induced conversion. But essentially what they're doing is they're developing a method to take that compost sample and see if it will infect other proteins and therefore still be infected. The hope with this research is to add a removal method as a tool for natural resource agents to help mitigate and control the spread of CWD. So if we can prove that composting is an effective way to reduce the infectivity of the prion or to remove the prion, hopefully they'll have something then that they can use in their management strategies.
The goal of this research, should it be successful, is a very practical, quick, easy, and safe way to dispose of the large amount of chronic waste disease infected deer that are being generated not only in Wisconsin and Michigan, but across the Midwest. This method is a really good way to capture the infection, hide it, trap it, and reduce the large volume of biomass that needs to be treated.
So being a part of Wisconsin, deer hunting and deer culture is a big part of growing up here and being a citizen of Wisconsin. And so we should care and manage these populations as best we can. And part of that is managing and mitigating the effects of CWD so that we can have viable hunts and have a viable deer population moving forward.